Okay, in chapter 5.2, we are going to talk about transport. We're going to talk all about transporters, so let's get into it. Okay, The Transporter is a series of movies starring Jason Statham. He drives a BMW in the first one, but then Audi paid him a lot of money to have Audis in the second and the third. Okay, not these transporters, right? Although classic films, not these transporters, unfortunately. We're going to talk about membrane transporters and how we move things across the membrane. So, we need to talk about two types of movement. We need to talk about passive transport and active transport. And this is very important. I will ask for these definitions on a test. Use these ones, please, and make sure you have them memorized and are able to write them out. So passive transport. Passive transport requires no energy because it is the movement of molecules across a membrane by diffusion. Remember, we talked about diffusion previously. That's from an area of high concentration to an area of lower concentration. That's what's causing the movement. That is why no energy is required, and passive transport will occur because of this concentration gradient. In some cases, though, we might want to move molecules in the opposite direction. We might want to move things from an area of low concentration to an area of high concentration. We might want to take the few things that are out there and pump them into the cell to have more of it, like things like food molecules, like glucose and stuff like that. We want to keep accumulating them. To do active transport, we have to spend energy. And in most cases, that energy comes from our energy molecule that is called ATP, adenosine triphosphate. So active transport is the movement of molecules across a membrane from an area of low concentration to high concentration, and it requires energy in the form of ATP. Okay, now that we know those two types of transport, we'll look at what we might be transporting in these cases. So, what do cells want to transport into them, and what do cells want to transport out of them? Go and pause the video, write some things down, think about this. All right, so let's start with things that probably want to go into the cell, right? Well, we have things like nutrients, okay, glucose, amino acids, lipids, other carbohydrates, things like that. Minerals, very important ions like electrolytes, and maybe even some gases, right? Oxygen, that's a big one that we need to get into our cells, right, for respiration to occur. All right, then what do we want to get out of the cell? Well, a lot of processes create toxic waste, so wastes and toxins, things like hydrogen peroxide, carbon dioxide, things like that. But we also might want signaling molecules to go outside of our cell. The cell creates the signaling molecule and it wants to send it to its neighbors. So we're not just getting rid of things we don't want. Sometimes we're getting things outside of the cell that we want to go to other cells. This is why we need to talk about transport, because it's really important in cell communication as well. In 5.2, we're going to start with passive transport. And I'm going to talk a lot about concentration gradients and some chemistry here because it is important for it. Passive transport doesn't need energy because it's using the potential energy in a concentration gradient. So there's a lot of potential energy in a high concentration. The molecules will flow down towards a low concentration. So we'll look at how passive transport occurs and why. We need to talk about a special process called osmosis, in addition to diffusion. Osmosis is a special type of diffusion. And then we'll talk about something that's called tonicity and has to do with the concentration of solutes inside and outside the cell. So let's go back to our basic chemistry concept here. We have the idea of a solvent. A solvent is a liquid that something can dissolve in. In many cases, this is water for biology. Then you have the solute. This is the solid that is going to dissolve in the solvent. Okay, so solvents dissolve things, solutes dissolve in solvents. Hopefully that's perfectly clear. All right, so let's look at an example here of diffusion. Remember, we said diffusion is the movement of molecules from an area of high concentration to low concentration. Here's an example. You have a glass of water and you drop some dye in there. The dye does not just stay in one place, okay? The dye is highly concentrated here, but it will start moving outwards by diffusion into the rest of the water. It's moving from high concentration out to the lower concentrations until it is all equal, right? All at equilibrium. So we looked at this example. We have a high concentration outside the cell, low concentration inside. That will move down the concentration gradient from high concentration to low concentration over time, and then they will equal out. Diffusion is one type of passive transport. This is called simple diffusion, where things move directly across the membrane. There's a problem, though. Only certain things can move across a plasma membrane. Plasma membranes are what we call selectively permeable. They allow some things to go through, but other things cannot pass through them. So what can go through them? It's a pretty small list. We'll focus on some big ones here. Generally, small non-charged molecules can diffuse directly across the membrane. That would be something like oxygen, right? A gas, or CO2. Water can move directly across. But when we talk about larger molecules like glucose, they cannot move across the plasma membrane. So we're going to have another way of getting them inside the cell. Other things that have a charge, like these ions here, sodium and chloride ions, potassium, hydrogen ions, they won't be able to move across either. So that's problematic, right? Because I've been telling you a lot about how we need glucose to live, right? So how do we get it into our cell? We'll come back to that in a moment. There is also one other class of thing that can move across here. There are some chemicals that are fat soluble, as we call them. There are some drug molecules like steroids and things like that that can move directly across this. Again, I'll reiterate, that makes them dangerous to handle without protection if you're not supposed to be taking them, because they can move directly through your cells, through your skin. All right, so what impacts how this stuff moves? Why will oxygen move in or out of the cell, or vice versa? Why will CO2 move in and out of the cell? Well, the biggest factor that influences the rate of diffusion, how fast diffusion across the membrane occurs, is the concentration gradient. The bigger the gradient, the bigger the high to the low, the faster diffusion is going to occur. Also, the mass of molecules is going to impact how quickly they move. Big molecules that are highly massive will move slower than smaller molecules. The temperature. Molecules will move faster when the temperature is higher, so this will impact the rate. Usually for biology, that means as things get cold, they slow down, basically. The chemistry slows down. Solvent density. Like if you are losing water, your cytoplasm technically becomes more dense. That reduces the diffusion rate in and out of your cells. One of the reasons dehydration is not good for you, among many. Solubility. The more nonpolar or lipid soluble a material is, the faster it will diffuse across that phospholipid bilayer. The amount of surface area. Coming back to this one, right? More surface area means you can diffuse things much faster. That's why we talked about the intestinal cells. Intestinal cells have all those little finger-like projections on there to increase surface area, faster diffusion. Then the distance traveled, right? Again, we said smaller cells, things diffuse faster in them. That's why we have limits on cell size. And then pressure. Pressure is a weird one in some organisms, in organs like the kidneys. Kidney cells, blood pressure forces things through the membrane, speeding up the diffusion rate. Okay, so back to our problem, though. We said big things aren't going to diffuse across the membrane. How do we get glucose into our cells so that we can power them, right? Here we have a neuron that is going to need a lot of energy. Well, this is where a special type of diffusion can occur. This is called facilitated diffusion. Facilitated diffusion uses transmembrane proteins, those integral membrane proteins, to move substances across the membrane. In some cases, they are like this little ratchet here, where they will ratchet back and forth and allow things in. In other cases, they're kind of tube-like. Here, we have some ions. Sodium, right, that's charged, and glucose, which is a large molecule, right? Your cells have a combination sodium-glucose transport system. This is why if you drink like a Gatorade or something like that, having glucose in there can help move the sodium ions in there, the electrolytes, because they move together. So the sodium and the glucose bind to this transport temporarily, and then it opens up and allows them in. Now, this is happening by diffusion technically. This is facilitated diffusion. The protein is facilitating the diffusion. We have a high concentration on this side, flowing to a low concentration over here. There is no energy being spent here, and this will stop if there's not enough on the outside of the cell, right? It will stop going into the cell. The membrane proteins shield the phospholipids from these charged particles and allow the big things to move through. There are other examples where it's just kind of a tube. Here's a transmembrane protein that is just a tube, and things will flow through it. Here we can see the hexagons have a high concentration on the outside, and they flow through the transmembrane protein to the inside where there's a low concentration. There's some examples here. We have things like aquaporins, which actually allow water to flow in and out of the cell depending on concentration. There are some of these things that have little gates on them that can open and close. Muscle cells have this on them. Our muscle cells fire by releasing a large amount of ions across these transporters. So they have to have little gates to allow that to happen when we want it to. But all of this is still governed by diffusion and a concentration gradient, right? So this is still passive transport. Channel proteins are not really interacting with the substance. Like I showed you with the glucose sodium transporters, there are some things like carrier proteins that actually do physically interact with the molecule that they're transporting. They bind to it temporarily to carry it across. But again, they can only move with the concentration gradient from high to low. These are generally very specific to their substance, so they're only going to transport one thing. In this case, the green ovals move through this versus the green hexagons do not. Again, we have things like kidney cells that have glucose carriers that filter glucose out of the blood and can concentrate it. So these are slower than those tube-like channel proteins, though. There is also an important point here. The more transporters your membrane has, the more stuff they can transport at once, right? You're limited by how many of these you have. So oftentimes it's not just one of these things. There's hundreds, thousands of them embedded in the cell membrane. All right, I want to talk about water specifically here. Water is very special when it comes to its movement. And when we talk about the movement of water, we don't talk about diffusion. We talk about a process called osmosis in a lot of cases. So osmosis is the movement of water through a semi-permeable membrane. All right, what the heck does this semi-permeable mean? Well, here we have a beaker, and in the middle, we have a membrane that will allow some things to move through it and other things not to. That is semi-permeable, right? In this case, we have the red liquid, that's water, and we have these purple spheres here. They cannot move across the membrane, but the water can. So you'll notice here, we have an imbalance in concentration here. Water is going to move to balance the concentration of solutes. That is the process of osmosis. So let's look at this. We have a high concentration here of purple spheres and a low concentration here. This chemistry, we would love for these concentrations to be equal. we could achieve that in two ways. We could add water to this side to make this less concentrated, or we could subtract water from this side to make it more concentrated until they're equal. Osmosis is going to do both things at the same time. The water is going to move from this side. It's going to move across to the other side. That will increase the concentration here and decrease it here until they are equal. Osmosis will only happen when water can move, but the solute cannot move across the membrane. This is actually very similar to what happens in cells, right? The membrane is semi-permeable. One of the things we said that could move across it was water. So osmosis is constantly impacting your cells. There is even a term called osmolarity that can be used to describe a solution's total solute concentration. Low osmolarity has more water to solute. High osmolarity has higher solute to water. So these terms are important when we talk about water balance in organisms. And that comes to a way to describe the difference between the amount of solute outside a cell versus inside a cell. And this is where we talk about tonicity. It describes an extracellular solution's concentration. So we're talking about what is the concentration of solutes outside of a cell compared to inside. That's what tonicity is talking about. So here we have an example. We have our cell here with its semi-permeable membrane. We have the liquid inside the cell, which has these red spheres at a certain concentration. And we have the extracellular solution, which has red spheres at a certain concentration. If the concentration outside the cell and inside the cell are equal, these are, outside is what we call an isotonic solution. Water will technically move back and forth between this, but it will move at an equal rate. So there's technically no net movement of water here. There is water moving in and out. There's always some motion in chemistry unless things are at absolute zero. Isotonic describes the outer solution when it's equal in concentration to the inside of the cell. All right, there's two other possibilities beyond isotonic. We have hypertonic, where the solution concentration is greater outside of the cell than inside of the cell. We'll see what happens when that occurs. Or we have the solution concentration is less outside the cell than inside the cell. That's a hypotonic solution. So hyper means higher. Hypo means lower or less. So it is lower outside. Okay, you'll notice in the video that I actually changed this as I was describing it. I want to reiterate that the hyper and hypotonic term is describing the fluid outside of the cell. So I think this terminology describes that best, right? Hypo, it's lower outside. Hyper, it's higher outside. All right, let's look at what happens to a cell when it is in these scenarios. Let's look at a hypertonic first. Hypertonic, higher concentration outside the cell than inside the cell. Well, the water in this cell is going to move outwards. It's going to try to dilute this here. It's going to try and lower the concentration outside, and by moving out, it will raise the concentration inside until they are equal. That means for a cell that a cell would shrink and shrivel under hypertonic conditions. Okay, it's not generally good for cells for that to occur. Let's look at the other scenario, though, the hypotonic, where it is less concentrated outside than inside. Well, in this case, the water from outside is going to flow into the cell. And that could be just as bad or worse. It could cause the cell to start to swell. And if too much water goes in, the cell will burst or lyse, and then it's game over. Your cell is dead. All right, why am I talking so much about this? Well, we obviously are worried about dehydration and things like that in physical pursuits and things like that. But We also have to think about this when we give IV fluids, okay? If you've ever got an IV, it contains all kinds of electrolytes, sugars, but it also is at a specific tonicity. So when we give IV fluids, we generally want them to be isotonic, equal to the concentration of solutes inside of red blood cells. Because if you were to give someone pure water, effectively a hypotonic solution, their cells would swell and burst. So distilled water would cause hypotonic situations. Red blood cells would burst. If you had too much salt, that would draw water out of the cells, causing them to shrivel. And shriveled red blood cells cannot carry very much oxygen. So here's another diagram showing that a little more carefully. Here's hypertonic solution. Water's moving out and the red blood cells shrivel. Isotonic, they are perfectly nice and round. Hypotonic, you don't have a cell wall, so your cells will probably burst here. We will actually examine these conditions in the lab. Plants have it bad because they need a lot of water, okay? And so they have evolved a cell wall that can help them deal with huge shifts in water. Under hypertonic conditions, water might move out of the cell. As water moves out of the cell, you've probably observed plants wilting, right? If you don't water your plant for a few weeks, it starts to wilt, right? An isotonic cell here will have equal movement in and out. But you come home and you find that your plant is wilted. What do? You pour a bunch of water on it. Well, now you're creating a hypotonic scenario, and water will rush into the cells. Luckily for plants, they have this very rigid cell wall that resists bursting here. It actually plumps up the plant cell, though, and we call that turgor pressure. And that will cause your plant to unwilt and start standing back up, right? So it's actually the pressure in the cells themselves. So let's think about what other organisms worry about water and osmolarity, the concentration of the water they're in. Any ideas? Well, you might be thinking things that live in water, marine organisms like fish and other things like single-celled eukaryotes like paramecium and amoebas that live in water and things like that. We have fish, right, that live in fresh water. That is a hypotonic scenario. Okay, hypotonic water is going to want to rush into the fish cells, so they're going to actually have to spend energy to pump water out. Same thing, opposite in a marine environment where we have salt water, right? There, water is going to be pulled out of the fish's cells. So fish actually spend about 5% of their metabolic energy just on regulating the tonicity of themselves and pumping water in and out and things like that. So osmolarity is a big problem for organisms, particularly those that live in water. All right, let's finish with a review question from last chapter. So here, the transmembrane domain of an integral protein interacts with the tails of membrane phospholipids. To do that, they must be which of these things? Go ahead and pause the video, think about that. To interact with the hydrophobic tails, they need to be nonpolar. So the transmembrane domain is often nonpolar here. All right. That's it for passive transport. In the next chapters, we'll talk about active transport. So remember, passive transport, movement of molecules across the membrane by diffusion from high concentration to low concentration. No energy required. Active transport, on the other hand, is movement of molecules across a membrane from an area of low concentration to high concentration. That will require energy. We talked about diffusion and its definition. Solvents are liquids that can dissolve something. The solute is the thing that dissolves there. And we talked about selectively permeable membranes and the factors that influence diffusion. We looked at some facilitated diffusion, integral proteins that can help move things by diffusion. Then we talked about the movement of water across a membrane, that is osmosis. We talked about osmolarity, the solute concentration in solutions, and tonicity, and how that impacts the cell. Okay, we'll move into active transport next.